Hey, welcome to Biology Carol and Tuesday afternoon biology, straight after Easter. Any of you who are out there listening to this, can I just say, well done. It's very impressive. So today we are going through an OCR A exam paper. We're going through paper one. So on the paper one, you've got 100 marks out of the total of 270, which ends up being about 37% of your whole A level. That's the same proportion that paper two is. Paper three is obviously, 26% of the total. So this is clearly a major chunk. So we are going to go through relatively quickly each of the questions on the paper. I'm going to show you what I would have written. I'm going to show you the mark scheme, and I'm going to show you exactly where I got the marks. I write the numbers of the mark points right next to the point at which I get the marks on the longer prose questions. But first of all, let's go through section A, which is obviously the joy that is a multiple choice. So, following statements, which of these correctly explains the feature of an efficient gas exchange? Layers are thin for short diffusion distance. I'm liking that answer. I'm kind of thinking, yeah, that's possible. Good blood supply reaches equilibrium. No. Increase surface area to reduce surface area to volume ratio. No, that's rubbish. Ventilation takes place to reduce a concentration gradient. Rubbish. It increases the concentration gradient, makes it steeper. So A, and that was the right answer. This one is about you understanding what hydrolysis and condensation reactions are. Hydrolysis splits bigger molecules into smaller ones. Condensation does the opposite. It makes bigger molecules from smaller ones. So we're looking at what is happening to the size of the molecules. Nucleic acid, big molecule, DNA, RNA, goes to a nucleotide, hydrolysis, that makes sense. Doesn't make sense for hydrolysis to be how it makes itself into a larger polynucleotide. So that is not correct. Alpha glucose, condensation to a myelopectin, yep, that makes sense. Hydrolysis back to alpha glucose, liking that answer. Amino acid condensation to dipeptide, absolutely right. Hydrolysis to polypeptide, load of rubbish. Beta glucose condensation reaction. I can't read the rest of it underneath here, I'm afraid. I can't remember, but I know that this is number B. So we got that one okay. So following table describes the approximate percentage mass. Percentage mass, this takes you back to your GCSE calculations, all those molar calculations of different chemical elements in organic polymers. So I looked along these and I was kind of going, well, this seems about right to me. Hydrogen is the lightest atom in the universe. So it makes sense that even though there's a lot of hydrogen, it's going to be a small proportion of the molecule in percentage mass terms. So I like the look of A. I didn't like the look of B because basically this hydrogen number is just far too high given that hydrogen is the lightest atom in the universe. Didn't like the look of C because proteins, amino acids, they're not 50% phosphorus. I kind of knew that. And lipid, this doesn't make sense because even though hydrogen is a very small percentage, it's too small. And it certainly wouldn't be nearly 50-50 carbon to oxygen. So I went with A, and that was the right answer. Which of the following statements about DNA replication is correct? So in terms of A-level knowledge, A is not correct. RNA does not bind to DNA, not that you learn that at A-level. The distance between the strands of the double helix will always be the same. Well, it will be in a normal double helix not undergoing replication, but of course, those two strands are split and therefore the distance isn't going to be the same. Adenine is not the same proportion of guanine, so that is wrong. And now I'm kind of going, I flipping hope that D is right because otherwise I'm stuffed. Phosphodiester bonds will occur in the same direction. Yes, they do. because of the directionality of the two strands of DNA. So here we go, that's D. Which letter A to D shows the rate of reaction with a fixed quantity of competitive inhibitor? Now, I just immediately know that the answer is B. A is the classic curve without an inhibitor, and B is the one with a competitive inhibitor, because at the high substrate concentrations, the rate of reaction is the same as it would be if there was no inhibitor in the mix. So I went for B, and that was the right answer. Which of the statements A to D applies to transpiration and evaporation? Okay, faster rate at higher humidity. Well, that's a load of rubbish, because the higher the humidity, the slower evaporation happens. You don't get your clothes dry in a tropical rainforest. Here am I thinking that evaporation, transpiration is basically what happens on a clothesline. So it occurs at a slower rate with a greater wind speed. Well, that's clearly a load of rubbish as well. It occurs at a slower rate at a higher temperature. Garbage. Okay. It occurs at the surface of the leaves. Got to be D. There we go. Can I just say, if you have any questions, do put them up in the chat. I am racing through this so that it doesn't end up being a tediously long drawn out process. If you have questions, put them up, but I'm running a good 10 seconds or so ahead of what you are hearing. So, you know, just be aware of that. But if I need to go back to a question, I will try and do so. Which of the statements A to D explains why diastole follows systole in a mammalian heart? I had to think about this one, because I kind of went with, you know, with A, I'm kind of going, oh, it's myogenic. Well, that's about how it synchronizes the heartbeat. So I'm kind of going, okay, I quite like that answer. B, cardiac muscle takes a short time to repolarize after being stimulated, to repolarize. In other words, go from, if you remember your action potential, go from hyperpolarization back to the membrane being polarized again. So I quite like that one. Aorta is capable of maintaining a pressure generated by, well, that has nothing to do with it. SAN receives impulses from the AVM. Well, that's rubbish. So it's out of A and B. And now I'm thinking, which one is the better of the two answers as far as I'm concerned? And this to me would be B, because it explains why contraction happens, systole, and if there is a short time to repolarize after being stimulated, well, that explains why relaxation follows systole. So I went with B, and it was the right answer. Okay, this is the classic Banting and Best experiment, which in our day and age sounds pretty horrific. And okay, it is pretty horrific. Okay, so what they did, they removed the pancreases out of healthy dogs, produced an extract called ilotin. Okay, just go with it. The ilotin, this extract that they made from the dog's pancreases, was then injected into dogs who had had their pancreases removed. And they tested the blood glucose concentrations while, you know, as a result of the injections of this ilotin extract. So which of the following statements and here's the graph, obviously, and here are the ilotin injections here and here, and you can see that the blood glucose concentration goes down immediately after an ilotin injection and then comes back up again. Have another injection, it goes down, comes back up. Which of the following statement correctly explains these results? Ilotin is made in the alpha cells of the islets of Langerhans. Well, I'm looking at this graph and going, this is insulin, because insulin reduces blood glucose concentration. So I'm kind of going, no, that I'm not convinced about at all. ilotin reduces blood glucose concentration. Absolutely, they inject it and the blood glucose concentration goes down. The effects of ilotin are short-lived. Now, this is a bit of a tricky one, I'll be honest, because there's no timescale on the x-axis. But I also know that the effects of insulin are short-lived. You know, our bodies are constantly having insulin secreted and glucagon secreted into our bloodstream. So I'm going with 2 and 3. And I can't now see, I've got a little window just at the bottom, permanently at the bottom of my screen about this whole live streaming thing, so I can't see the answer. So it is C. Fantastic, I got that right. Calculation. Oh my word, here we go. Which of the following is the percentage increase from the PEF, peak expiratory flow, whatever, of a 20-year-old man, right, 20-year-old man of 175 centimeters, middle line, 20 years. That is 570 because it comes over here. Okay, to 45-year-old man at 83. Well, that is here, so that is 640. Now, percentage increase. We are going from a shorter step up to a bigger step. And we want to know, here is our increase, here is our increase, and we need to know the percentage increase relative to the original value, the original amount. So our increase is 70. And that we are now measuring that as a percentage of the original amount because we've gone from a lower amount upwards. So we've gone from 570, added 70 to that. So we're working out what percentage 70 is of 570. And that is 12.3, which is C, which was correct. So here we have a question about thyroid and thyroid cancer and iodine 131, radioactive iodine. Injected, or sorry, not even injected, I think it's just given to the patient. Given to the patient, it congregates in the thyroid because the thyroid is the place that uses iodine. And because it's radioactive, it kills the cancerous cells, but then it is excreted. And the question, and this graph shows you how is it excreted. Which of the following A to D? Oh, we've got a load, just realized I've got a load of questions. Here we go. Hello, Lavinia, nice to see you. Habib, I'm a GCSE student. How do gametes carry one allele? Okay, really quickly, Habib. Gametes only carry one allele out of the 2 that are carried for every gene. In every normal cell, apart from gametes, every cell carries 2 alleles of each gene, 2 copies of each gene, because you've got one from your mum and one from your dad, because the mum's egg, which only had one, combined with, fertilized with 1 sperm from your dad when fertilization happened, the first diploid 2N cell was produced. So why do gametes only have one allele? Because to make gametes, the process of meiosis has to occur. And meiosis is a form of cell division that reduces the amount of genetic material down to half in the gamete. So instead of carrying both chromosomes, because chromosomes come in pairs, instead of carrying both chromosomes, each gamete only carries 1 chromosome, either the one from mum or from dad. So that's why gametes only have 1 allele or 1 copy of each gene, because then when they fertilize with the other gamete, the chromosome number comes back to the normal again. Hopefully that's helped. But how do 50% of Garrett's clamp? Well, that's, yes. So if in meiosis, if a person's genotype is big A, little a, in other words, they're heterozygous, because each gamete only is going to carry one chromosome and therefore one allele, by definition, half of the gametes will be big A and half of the gametes will be little a. I hope that makes sense. Oh, just as if, thank you very much. Okay, all right. Hey, oh, Habib, can I just say, this is really full on if you're following this now. Take all of this with a pinch of salt or go away and have a cup of tea. Anyway, right, back to dear old A level and radioactive iodine being excreted. Which of the following correctly explains I-131 in each body fluid? So I'm reading these and I'm kind of going, okay, I quite like A. I-131 is able to cross capillary walls. Well, it will be, but I'm not. I'm just kind of going, I'm not convinced about that as an answer at all. Kidneys are more efficient at excreting 131 than the lungs. And I'm going, that is definitely true. Does it explain all of them? Because this, in each body fluid, I'm kind of rereading the question. I'm kind of going, not sure that C is the right answer. And now I cannot see option D, but I know I discounted it. Something about thyroid. Really sorry, I just can't see it on my screen, which is very irritating. Oh, hold on, I can read it here. Well supplied with blood. Yeah, not convinced by that. No, load of rubbish. So I'm choosing between A and C. And to be honest, I lumped for C, but I found this one tricky to know which was the right answer. I got it right. I'm going to be honest, it was more luck than judgment. Which of the following A to D is similar in the way ATP is made in respiration and photosynthesis? Both involve NAD. That is wrong, absolutely, because photosynthesis involves NADP. NADP, remember P for photosynthesis. Both involve substrate level phosphorylation. No, because that only occurs in respiration. Substrate level phosphorylation doesn't occur in photosynthesis. Both involve, now when I first read this, I read it as protons. So I went, oh yeah. And then I read, both involve proton gradients. I'm going, how am I supposed to choose between these two? And then I reread C and realized that it said photons, not protons. So photons are packets of light energy, and therefore, obviously, only photosynthesis involves photons. Both involve proton gradients, hydrogen ions, absolutely. D, definitely. Okay, image below of the human brain. Which of the labelled regions would be directly involved in learning to play a musical instrument? Clearly, only 2 out of the 4. And I'm immediately going, well, you're learning something that is definitely going to be involving, you know, the cerebral cortex, which is Z, and it is also going to involve the cerebellum, because if you're learning a musical instrument, those are fine motor skills and fine coordination. So I would definitely go for W and Z. X is the medulla oblongata, and that is only involved in controlling heart rate and breathing rate. And Y is the hypothalamus, and that is, you know that for being detecting changes in the blood, for example, temperature and water potential. So W and Z, which is C. So which of the following diagrams is a sensory neuron? It is D. B is the motor neuron. A, I have no idea what they're trying to show, made up cell. C is clearly a ghost. So there we go. All right, table below, load of potential differences across membranes. Oh, here we go. More questions, sorry. Could you do ASAQA biology paper, please? I'm going to be honest, until the end of the year, I'm going to be concentrating on A level rather than AS, I think. Sorry. Right, you have a question on bore shift. There isn't one on this exam paper. I know that, Lavinia. I've done a whole video on bore shift, which is on my website, www.biologycarol.co.uk. So do tap that into Google and go and find that. I've got videos for the whole of AQA and OCR A syllabuses now, and I sell the whole lot for 50 quid for a year subscription, which is peanuts for hundreds of hours of teaching. Well, certainly what there would have been at school. I condense it down, make it fast, and I go through exam questions as well. So go and have a look. I think that's probably the best thing. So just to summarize, this is what this is talking about. You've got 3 synapses joining with a postsynaptic neuron. And they're telling you what is happening and whether the postsynaptic neuron is fired. -70 means that it's staying at resting potential. So number 1, A is firing, postsynaptic isn't. Number 2, B is firing, but the postsynaptic isn't. Number 3, C is firing, postsynaptic isn't. Number 4 is A and B are firing, postsynaptic isn't. The only time that the postsynaptic neuron fires is when A, B, and C all fire at the same time. That is spatial summation. Temporal would be if two action potentials came down one neuron in very close succession, very quickly, one after the other. Hyperpolarization doesn't apply. Divergence is if you have one neuron going off and having a synapse with several others, and so that doesn't apply either. So it is C. Drug metoprolol prevents the stimulation of postsynaptic receptors in the sympathetic nervous system. Right, sympathetic nervous system speeds things up. This drug prevents stimulation of the speeding up nerves. So metoprolol slows things down. So which of the following conditions would this drug be used to treat? Well, muscle fatigue is when muscles are relaxed, they can't contract. They're not working well. You don't need to slow them down anymore. Tachycardia is a fast heartbeat, which you've got to know that term. So definitely 2. And high blood pressure is when it's partly due to either the heart rate going too quickly or the arteries particularly being constricted. So, and therefore too much muscle contraction. So I would go for 2 and 3, but not 1. So C. Section B. Gestational diabetes affects pregnant women. Gestation means basically pregnancy. High levels of blood glucose in the blood, even though, here's the crucial thing, even though the woman produces normal levels of insulin. Which type of diabetes is gestational diabetes more similar to? And that is type 2. And here am I answering this. I didn't know whether type 2 would actually get me a mark. So I'm then giving 2 reasons why. Number 1, because this is a 2-mark question, insulin is still being produced by the beta cells of the islets of Langerhans, because I don't quite know how much detail to put in. And the body cells don't respond to the insulin. So here is the mark scheme, and actually I ended up getting 3 marks or hitting 3 marks. Obviously I'm only going to get 2 because it's a 2-mark question, but I got all of those 2 marks. Probably worth you knowing that when I went through and did this paper, I deliberately didn't look and I can only tell you, I didn't look at the mark scheme as I was writing my answer. I then obviously put up the mark scheme and then marked it. So I've been as honest as I can be. Oh, crumbs, I've got loads of marks there. Anyway, right, so just 2 ways a woman can manage her condition. Well, because it's blood glucose that's the issue, it's eating less carbohydrate, controlling the carbohydrate intake. And the other classic one is exercise, which I just kind of know. Notice your list rule. If you had written both of these answers under number 1, they would have just taken the first 2 answers that you wrote under number 1. If you wrote 2 answers each under number 1 and number 2, they would have simply taken your first answer from number 1 and your first answer from number 2. You will never get away with giving more than 2 answers when they only ask you for 2 answers. Because does it make sense? If you provided 10 answers, really likely you're going to hit on the correct 2 answers. So they just make sure that you can't do that. Okay, so skeletal muscle, one of the main tissues where glucose is removed from the blood. Yeah, where's the other one? And the other one is the liver. Oh, I got those 2 marks. Yes, sorry. And the other one is the liver, which I got. Explain why glucose is required for the contraction of skeletal muscle. So here am I starting with glucose. And I'm thinking of the stages that have to happen. This is a 3-mark question. The stages that have to happen to make muscle contraction occur. So immediately I'm kind of going, well, glucose is required for aerobic respiration so that ATP can be produced, because now I'm at the point where I can use my ATP for the muscle contraction. But now I'm thinking, what do I know about how ATP is used in muscle contraction? Well, I know that the role of ATP, specifically in muscle contraction, is to break the cross bridge of the myosin head and the actin molecules. So I wrote that, as opposed to just saying, and ATP is used for muscle contraction. So hence I got all 3 marks. So notice that I can say legitimately that ATP is produced because ATP is a chemical. You can never say, as is rightly written here, you can never say that energy is produced, because energy is neither created nor destroyed. So you cannot say that. You can only release energy, but you can produce ATP. So I got all 3 marks. Okay, during late pregnancy, they did this study in a woman who was 36 weeks pregnant. A pregnancy only lasts 40 weeks, so this woman was about 8 months pregnant, heavily pregnant woman. And here is her spirometer trace. And you're told that the mean oxygen uptake of a woman at rest is 0.02 decimeters cubed per second. I go, okay, all right. Using these data, in other words, this one and all of this, the student made the following conclusion. My data show that being pregnant reduces the rate of oxygen uptake by up to 20%. And you've got to now say whether this is true or false. So first of all, I'm kind of going, oh, flipping heck, I've got a big calculation going on here. And I have got to work out the rate of oxygen consumption. Well, we do that by working out the gradient drop, or the reduction over time of of this line here. I'm not going to go into how a spirometer works. I haven't got time for that. So I kind of looked at this, and I quite liked going between 10 and 70 for 1 minute, taking in most of the plot. I just, so I kind of went, okay, at 10, we've got 2 decimeters of air, and by 70 seconds, it had gone down to 1.25. So that means that the difference here is the oxygen consumption, which is 0.75 decimeter cube per minute. Of course, they want it, for me to be able to compare these numbers, I've got to make it per second. So if this is how much it is in a minute, I've got to divide this number by 60 to get it as per second, which was this. But now I've got to work out, it reduces it by up to 20%. So I've now got to work out a percentage reduction. So I've got to see whether going from 0.02 to 0.0125 is either less than a 20% reduction, percentage reduction, or over 20% reduction. So here is my calculation. So we have got 0.2, here's 0.02, and this is 0.0125. So the difference is this bit here. But we have got to work it out as a percentage of the original number, because that is the percentage decrease. So it is 0.02 minus 0.0125. There's your decrease. But to make a percentage decrease, you put it over 0.02 and times it by 100. And I've got a 37.5% decrease, which is clearly greater than 20%. Now, by the time I'd done all this calculation, I'm going, flipping heck, I think I've earned my money for these 3 marks. And I didn't write anything else. Now, I had shown that this is actually an incorrect statement. So, but it is as far as I went, because I really thought I'd done enough. And as it was, looking at the mark scheme, I had got mark points. Oh, I'm going to come to questions in a second. Mark points 1, 2, and 3. But I want to point out to you that absolutely legitimately, we could have critiqued this methodology. The fact that only one woman had her spirometer trace taken, and that there's only one woman, and she was really heavily pregnant. And this statement is talking about, you know, being pregnant. Well, it's just far too broad a statement from the experiment that was carried out. And there is your classic technique in evaluating data. Look at the data and see if you can find things that either agree or disagree. Say both if you can see both. And then start critiquing the experiment itself, because almost certainly this statement will not match up with the experiment that has been done. And you can talk about number of repeats like we have in this one. Okay. Oh, Hussein. All right. T-Boy, nice to see you. I'm good, but I'm in a rush. I'm trying to be efficient, so I'm going to keep going. All right, question 17. Chromista. Photosynthetic protoctists. Oh my goodness. Protoctists, let me just tell you, protoctists are the rubbish bin of classification. Animals and plants, both multicellular. Fungi, multicellular. Bacteria, all of these other 4 kingdoms, bacteria, single cellular. Protoctists, all single-celled organisms, but some will be more like animal cells and some will be more like plant cells. This one is like a plant cell, a unicellular plant cell. Okay, if that makes sense. And it lives, so it's a protoctist and it lives in water. Okay, weird, here we go. They contain chlorophyll C, and chlorophyll C isn't found in plants. All right, fair enough. Outline the importance of photosynthetic pigments in photosynthesis. Okay, I had to think for a couple of seconds. The importance of photosynthetic pigments. Well, the number one thing is that photosynthetic pigments absorb light energy. Number one. And that causes the release of electrons from the primary pigment, photoionization. And that is so that those electrons can go down that electron transport chain so that ATP and NADPH can be produced in the light-dependent reaction for the light-independent reaction so that photosynthesis can occur. And you're just kind of going, oh my goodness, how much information do they need or want to get the marks? And I kind of said all of that, as you can see in the first part of my first part of my answer. And then I had a few lines spared. So I went back to the question and went, I haven't talked about accessory pigments. I'm going to say that they pass the light energy down to the primary pigment, you know, so that a greater range of light wavelengths can be used. And here we go. So I got mark point 1, mark point 3 down here about accessory pigments. This is mark point 1, mark point 2, mark point 3. Here is mark point 4 because this is about primary pigments losing electrons to go down the electron transport chain. So there is my mark point 4, and that was for the light independent reaction. So I got all 4 of the marks. Sorry, suggest why chromists need pigments that are different from those of. Now, I felt like I was scraping the bottom of the barrel here because I realized that they were in water. So I just kind of went with, hey, not all wavelengths reach down into water. That isn't something that is really on your syllabus. And this is the mark scheme. And can I just say, hardly any students got this mark in the exam, very few in the examiner's report. So just useful to put that in context. This first one as an option, now I got the mark point, and in fact it isn't mark point 1, I actually got this mark point here. This one I would never ever have written. And it is true that as light passes into water, the wavelengths shorten. It's kind of one of, it's one of the reasons why, and then it's reflected, and it's one of the reasons water looks blue. You know, it's because, it isn't absorbed, it's reflected. Yeah, it's slightly different anyway. But, you know, I'd have never, ever have written this. And this, I really felt like scraping the bottom of the barrel. But as I say, very few students got this. So please don't beat yourselves up for not knowing what to write here. So here we've got a chloroplast in a chromister cell. They've labelled the external membranes, they've labelled DNA, they've labelled thylakoids stacked in 3s. Slightly weird. Okay, notice that. Always read all the information, it gives you clues. Outline the structural differences between this chloroplast and chloroplast in flowering plants. So the two things so here we are looking at 2 things. 2 things that were obvious to me was that number 1, in fact, the most obvious thing was that you didn't have these little interconnecting lamellae between the thylakoids. Notice that I was very careful to say what the chromister chloroplast was like and what flowering plants chloroplasts were like, to really nail the marks so that it was very clear about how I was comparing the 2. And I used this clue here for my second point, that they only have 3 thylakoids per granum. Flowering plants have many more thylakoids per granum. So, and I got, what did I get? Mark point 2 and mark point 3. Okay, here is a diagram of the plasma membrane of a chromister cell. And notice that A is not indicating the whole phospholipid bilayer. It is only indicating the hydrophobic tails of the phospholipid bilayer. State and explain how one property of region A, only the tails, contributes to the stability of the plasma membrane. Now, I'll be honest, the only thing that jumped out at me here was the fact that this part of the phospholipid bilayer has cholesterol molecules in it. and they bind the hydrophobic tails together, and therefore the more cholesterol molecules you have, the less fluid the membrane. And that's what I put. So suggest has cholesterol molecules, explain, because the less cholesterol it is, the more fluid the membrane, or the more cholesterol, the less fluid. And therefore that, and I probably should have put therefore, you know, I think I should have put really, therefore making the membrane more stable. So I got mark points 3. Notice here, ignore stability for explanations. So if I had just said the less cholesterol, the more stable the membrane, I wouldn't have got the mark, because that was kind of in the question. So they were just kind of going, no, give us something more than that. And the property must be linked to the explanation. You couldn't just, you know, talk about something else random. You had to link the two. So I got mark points 3 and 4. Differences between plasma membrane and the membrane within cells. The plasma membrane is the cell's surface membrane. Outline the roles of membranes within cells. Well, immediately I'm thinking membranes within cells allow compartments, allow compartmentalization of the cell. And the obvious thing for me was to say that, you know, that allowed the formation of membrane-bound organelles, for example, Golgi and RER. And the other most obvious thing that I could think of that wasn't related to compartmentalization, because I didn't want to kind of make the error of talking about two things that were so similar they ended up being the same mark. And therefore I wrote that enzymes and proteins are embedded in the membrane, and that is another, you know, another role, as it were, of the membrane within cells. And I gave the example, you know, the electric transport chain and ATP synthase. And so therefore I got mark, but in fact I should have put, yes, I got mark point 1. In fact, I didn't get it there. I got it here. Yeah, because this is the end. Oh no, I did get it, compartmentalization. Sorry, I'm talking rubbish. Okay, and then I got this one here, mark point 4 as well. 1, 2, 3, 4, yes. Okay, question 18. Plants lose water by transpiration. You can measure it using a potometer. Plan an investigation into the rate of transpiration in 2 species of plants. That's the independent variable that would allow for valid data to be collected. That's talking about all the control variables. Notice details of how to set up a potometer are not required. You would be wasting the time and your lines in writing anything about that. So I talked about setting up a potometer, one with species 1 in it with a stem of species 1, one with a stem of species 2. Then measure over a controlled time period the distance moved by the bubble in the potometer. I didn't have to explain about the potometer, didn't have to talk about the setup, so I've just kind of gone with it. From this movement, a rate of transpiration can be calculated. And literally, as I am writing these words, I'm thinking about how can I pack as much of my knowledge into this, because here now I'm indicating that it is a volume of water that I'm calculating, because of course it's the volume of the cylinder that makes up that capillary tube, that bubble moving down the capillary tube, divided by time. I'm telling the examiner that to work out a rate, I know I have to divide by time. And I should have put here, I'm going to be honest, I should have put time taken to use up that volume of water. Now I'm hitting the valid data bit of the question. Variables to be controlled: temperature, wind speed, humidity, light intensity, because of course light intensity will cause stomata to close. And I was about to write leaf surface area, and because I know my biology, I kind of thought, hold on, you can't control leaf surface area, particularly if you're talking two species of plants. You could have completely different, leaf surface areas. So now I'm really going into the detail and kind of saying, okay, the surface area of the leaves of each of the plants would need to be measured. And I could have just put in brackets using graph paper. To work out an area of an irregular surface, you would use graph paper and count the number of, little boxes, so that the rate could be calculated. And I've just realized I've written this wrong. This should say per centimetre squared of surface area. Okay. And then also about the validity, you've got to repeat, you've got to have numerous repeats of your experiment so that a mean can be calculated, an accurate mean can be calculated. And I could have put also that anomalies could be identified. So notice we've got an asterisk here. This means that it is one of these quality of written communication questions. So it's going to be leveled. Teachers or examiners have to read through the whole answer first, gauge how much science content there is in the answer, gauge whether it has been written as, continuous prose, it's been written logically, and all the rest of it. So, but the science content determines the level, and the communication statement, this one in italics, determines the mark of that level. So, and here is an indication of the science content. Notice these are not mark points. I'm not going to go through my answer kind of going, oh yeah, got that, and count them up and go, oh, I've got 6 marks. Not how these ones work. So I've written, I think, a pretty full and detailed plan as to how to carry out a valid investigation, because I've included control variables. And here's my communication statement. I think it's a well-developed plan, shows the appreciation of need to contain valid data, and it's relevant and it's clearly explained. I haven't gone off on one about something else. So I think I would have got 6 marks for that. Plant cells walls made of cellulose. Cellulose polymer of beta-glucose give 3 properties of cellulose that make it suitable as the basis of plant cell walls. And here are my answers. And here is the mark scheme. And I looked at the mark scheme and I went, oh, I've only got 1 out of 3. And I didn't quite understand it. And I had a look at the notes, looked at the list rule, yeah. And it said, ignore permeable, ignore strong. And then it dawned on me that what I have written here, so I've got 1 out of 3, not great, but here I have written about the properties of cell walls, not the properties of cellulose. We're talking about the property of the molecule, not the property of the structure that it goes on to make up a large proportion of. So that was, my bad. Okay, here we go. Explain whether the action of these, so bacteria in stomachs produce enzymes that can digest cellulose. Explain whether the action is intracellular or extracellular. Well, bacteria are single-celled organisms. They are secreted, they produce the enzymes. So those enzymes are extracellular to the bacteria because the bacteria have secreted them. So this is what I put. Again, I wasn't sure whether extracellular would get me a mark. Here is the mark scheme. And I've actually given myself 2 marks. That's a load of garbage. This should have just said 1 there. So it's extracellular. So you didn't get the mark for saying extracellular, but you had to include it. You had to include it because the action takes place outside of the bacterial cell. So yeah, and notice secrete and excrete are two completely different processes. Secrete means basically pushing a liquid out of a cell, pushing a substance out of a cell, and excrete means getting rid of metabolic waste. So they're completely different processes. Honeypot ants belong to several different genera. Okay, all right. Okay, oh, lovely, swollen abdomen, very swollen abdomen. Investigation carried out into the respiratory substrates of 3 different genera of honeypot ants by measuring oxygen uptake and carbon dioxide production. Well, I'm immediately kind of going, this is respiratory quotients. And they've told you, fill in the table. And they've told you that it's the first answer, mainly carbohydrates. So I'm kind of going, right, I'm going to do the respiratory quotient. And that is CO2 produced divided by oxygen consumed. And the respiratory quotient, I know, is 1. And here we go, we've got 1.01. So this one, I then was on a bit of a roll, worked out the respiratory quotients, obviously, as I've done here. And I know that proteins have a respiratory quotient of about 0.9, funnily enough, and lipids have a respiratory quotient of about 0.7. So, and I followed the wording in this box here. So I just did mostly protein, mostly lipid. So, and I got the marks. Okay. Chitin is a polysaccharide found in insects. It's used to make the exoskeletons. Using information from this diagram, state 2 similarities and 2 differences between chitin and glycogen. You've got to look at this diagram. You've got to look at this relatively carefully. So I'm immediately looking at this and kind of going, well, they're both made up of hexose monomers. Here's one hexose sugar, here's another one, and these brackets and an N here says this just carries on and on and on either side. We've got many of these units strung together. So first similarity, both based on hexose monomers, as glycogen is, because glycogens are based on alpha glucose. And I notice here we've numbered these, so I'm going, right, I'm really confident that they're both 1,4 glycosidic bonds, because I know that to be the case with glycogen. And then I'm looking down here and I'm kind of going, well, hold on, flipping glucose doesn't have this here. So I wrote chitin has this group. Now I know if you've got H on one end, you have OH on the other, as far as glucose is concerned. So I've written chitin has this group on it, whereas glucose has OH. And notice that again, with a difference, I've clearly said what one is like and what the other is like, because that is one difference. And also I'm noticing, oh, this is just going on. This is a linear molecule, because it tells you that by saying that this is just repeated many times. It's a linear molecule. So I've said that glycogen is branched, whereas chitin isn't. And I got these mark points on the mark scheme. Mark points 2 and 4 and 6 and 8. The others are true, but they were less obvious to me. Okay, another quality of written communication question because of this asterisk here. Outline the processes involved in the generation of ATP through chemiosmosis. Notice chemiosmosis is emboldened. They only want you to talk about chemiosmosis. Don't need anything else. This is a 6-mark question, but this is fundamentally an AO1 question. It's regurgitation of knowledge, but they're wanting all the detail. So here we go. Chemiosmosis occurs as a result of electrons, and here am I putting in the source of those electrons. Those electrons have come from reduced NAD and reduced FAD. I did write this in, maybe I should have written this in full, longhand. Going down the electron transport chain. I know that ETC is okay for electron transport chain. Energy is released at each stage and is used to pump hydrogen ions across the membrane into the intermembrane space. This creates a concentration gradient, or it creates a buildup of hydrogen ions in the intermembrane space. And they can only diffuse through ATP synthase back into the mitochondrial matrix, because I've suddenly realized here, I haven't even mentioned this is in the mitochondria. I'm kind of going, oh, duh. Okay, as the hydrogen ions pass through ATP synthase, ATP is produced. And whenever I talk about ATP being either produced or hydrolyzed, I always write the equation, because I'm aware that that is often a mark point in itself. And here I am really nailing the fact that the electron transport chain and ATP synthase are located on the inner mitochondrial membrane. So once again, as always, examiners have to read through the whole answer. They have to assess the amount of science content in the answer, and how full and detailed the description is. But whether I get the higher or the lower mark in the level depends upon the communication statement. And, to be honest, I think I've put in enough science content. Well-developed line of reasoning with accurate descriptions of the processes. The information presented is relevant and clearly outlined. I haven't gone off on one. If I had talked about the whole of what happens in the mitochondria, link reaction and the Krebs cycle, as well as, chemiosmosis, I would have lost a mark. If I had written it all beautifully, I would have lost a mark because not everything was relevant. Okay. So here I've highlighted the bits there. So, and I've included, notice these are not mark points. You don't just get a mark for everything you write, like on normal questions. But I think I've included a lot of these, or if I haven't used the term, I've described it, proton motive force, for example. So I think I would have got 6 there. Okay, question 20. Hey, Medrock. I like the name. Okay, I'm Jaded Jade. Okay, yeah, dear old Jaded Jade. I just know her as Jade, funny enough. Okay, delighted that you're here, and please do spread the word, Medrock. Excuse me. Student carried out an investigation into the effect of ethanol on the permeability of membranes in beetroot. Here is the method. Okay, which you've probably either done in the lab, I hope you did it in the lab last year, or if you've had a particularly sad 2 years, you've seen it on a video. Each step in the student's method relies on certain assumptions. For each assumption, here's the assumption, select the numbered step from the student's method that relies on that assumption. Okay, so which bit of the method is put in place as a direct result of this assumption? So, assumption A, pigment will only leak into the solution if the membranes are disrupted. Now, so here are my answers. 2, absorbance is proportional to concentration of pigment. Well, that is because we're using a colorimeter, yeah, that is the step that directly relates to this. Pigment will be released when the beetroot is sliced. Now, I came to this one and thought, oh, hold on, I've put 2 in already. I'm really sure that this is correct. So I went back to this one and looked at it again and kind of went, oh, hold on, 3 is. Because That is why this experiment works, because the ethanol disrupts the membranes, and therefore differing concentrations of ethanol cause differing amounts of pigment to leak out. So I changed this to 3, not being at all sure that putting 2 tubes would be the right answer. And blow me down, here is the answer. So I got the 3 marks. Student kept the ethanol solutions at the concentration. State 2 other variables that needed to be kept the same. Now, these were the 2 that I wrote. And that was, oh, I can't read. Oh, time allowed to wash the pieces in running water. Because here, what you're basically going is going through this method and kind of going, what haven't they specified? What haven't they specified? It all looks fine and dandy, but actually they've missed stuff out. So same species of beetroot. Now, I did feel like I was dredging the bottom of the barrel. When I saw the mark scheme, I kind of kicked myself a bit, because of this mark point 2 here, mark point 2. Equal sizes of beetroot, of course, do not mean that they're bound to have the same surface area. And, you know, and I kind of kicked myself a bit for that, because of course surface area is going to be something that hugely affects the diffusion of that pigment out of the cell. I mean, I got the 2 marks. Notice the list rule. Notice what they told you to ignore. Because obviously temperature had always been stated, time because it had been stated, concentration of ethanol, they'd said that there were different concentrations, and ignore size of beetroot wouldn't really make any difference. So I got the 2 marks. Okay. Here he's shown the graph plotted by the student. Make 3 criticisms. Now, I'm going to be honest, these were the 3 ones that I noticed first. I noticed the first thing I did was go along the x-axis and the y-axis to check that the numbers were all there. 0.6 is missing here. So I then kind of went, well, it should be, I first of all thought it should be a histogram, not a bar graph. You know, these columns should be joining, because these numbers theoretically are sequential. They're joining one onto the other. And then I kind of went, no, hold on, we've got way more than 5 points. This should be a line graph, not even a histogram. Okay. And the other thing I thought was that it should have a title. And again, I kind of think that possibly this was a bit obvious and I missed it. This hasn't got a flipping concentration. This probably should be, you know, percent, something like that. I got the marks, but I didn't feel like I got the most obvious marks, if I'm honest. Okay, hi, Shaq. Revising osmoregulation at the moment, but saw your life and just wanted to say, oh, you're very sweet. Thank you for the thank you. I do appreciate that. All my students used to laugh at me whenever they thanked me for a lesson. I would just kind of say, well, thank you for the thank you. And they'd just kind of think I was going mad. But anyway, right. Explain how carrying out replicates would improve this investigation. Oh, classic, common question. So replicates, notice that carrying out replicates help you identify anomalies. All right, because obviously if you do loads of experiments, you can see if you get a weird result. And you can then exclude those anomalies from calculating an accurate mean, because obviously the more replicates you do, the more accurate your mean is. So here we go. Allow you to anomalies to be identified and to calculate a mean. So I got mark point 1 and 2, I think. Yes. Okay, so don't allow prevents anomalies. Replicates don't prevent them. the more replicates you do, you're bound, almost bound to get an anomaly somewhere. Yes. And right, ignore, remove anomalies. That was probably because the important thing was about you calculating a mean. It isn't, you don't do replicates so that you can remove them. You do them so you can identify them, yeah. Okay, 21. Blue-ringed octopus even, most venomous of all animals. I've seen YouTube videos or Instagram or something, videos of people having one of these things crawling across their hands and them not realizing that it could actually kill them. Weird. Anyway, so it has TTX, which causes paralysis because this is what happens. Okay, binds to the external surface of a voltage-gated sodium ion channels in the axon membrane, and that means that the channel doesn't open. Using the information, explain how TTX affects the activity of neurons. Well, if it binds to the sodium ion channel and they can't open, it means that sodium ions can't diffuse into the axon. That means the axon doesn't get depolarized. That means that an action potential isn't produced. That means that the action potential can't travel down the axon. And therefore, and this was my weakest line here, therefore the stimulus can't be perceived by the brain. That's what I wrote. 4-mark question, and I got mark points 1, 2, 3, 4 and 5. Yes. Yeah, I didn't write 3, and I didn't think of writing 6, but I got all 4 marks. Right. Common cause of death is suffocation because of paralysis of the diaphragm. So if the diaphragm can't move, that means the diaphragm can't, so I just said it can't contract and relax, so the volume of the lungs can't change. That means the pressure of the lungs can't change, and that means the air won't flow in or out. Maybe I should have put or out either. But clearly the suffocation bit is if you can't get air in, even though it has, yeah, anyway. So I got, and oh yes, and I talked about the volume of the lungs and they refer to the thorax, but I'm okay because it's here. So I got mark points 1, 2, and I actually got mark point 3, but obviously only would have got 2 marks from there. Also known to reduce the speed of conductance in Purkinje fibers in the heart. Suggest and explain what effect this would have on heart rate. Well, so I'm thinking that so it slows down the speed of conduction of the Purkinje fibers, and those are the ones that are going down into the apex of the heart and coming round the side. So my logic was that that was going to slow down the passage of the impulse from the AVN, obviously goes through the bundle of His, then the Purkinje fibers. It's going to slow down the transmission of the impulse from the AVN to the ventricles of the heart. And therefore it's going to slow down the heart rate. So here is the mark. So here's mark point 1, slow down heart rate, causes ventricles to contract, and therefore got mark point 1 and mark point 2. Now, I didn't mention systole, diastole at all. I just talked about ventricles contracting. Now, I gave myself mark point 4, this one. Therefore, there's an increased delay. Yeah. But when I looked at the notes, ventricular must be mentioned once. Now, they haven't underlined it here, but I wasn't sure that I would get this mark because I haven't used the word ventricular, even though I've talked about ventricles contracting the whole time. Anyway, so I didn't. Hey, Elijah. Hi, nice to see you. I didn't get. I crossed out that mark, so I got 2 out of 3 there. Oh, and I'm sorry, I've gone on too far. Why is the transmission of action potentials along the axon slower in the absence of saltatory conduction? Well, that's because if it can't jump, the depolarization, which is the depolarization kind of literally leapfrogging sections of the axon, it means that depolarization has to happen along the whole length of the axon, and that is much slower. And that's my go-to answer. So whole axon needs to be depolarized. So I got mark 0.3. Okay, we're getting there. Scientists use a respirometer to measure rate of respiration and photosynthesis, okay, of maize in different light intensities. Okay, slightly weird. Scientists placed 10 maize seedlings in a respirometer, kept it in the dark for 3 hours. Only respiration going on there, not photosynthesis. Respirometer contains soda lime to remove any CO2 produced by the seedlings. Okay. The scientists placed 10 maize seedlings in a separate respirometer without soda lime and placed them at different light intensities for 3 hours at a time. So we've got 10 in the dark, 10 in the light. The ones that are in the light don't have the soda lime. Okay. The ones in the dark would have, that is a classic respirometer being used as a respirometer. Okay, first question. Okay, this is those classic, let's work out the volume of the capillary tube, the distance of the bubble move. Diameter of the capillary tubing is 0.1 millimeter. Volume of a cylinder is pi r squared times L. Calculate the rate of oxygen uptake by the seedlings in the dark. Now we know this happened in 3 hours. All right, so let's make that clear, in 3 hours. Give your answer to 2 significant figures and show your working. So here am I drawing the capillary tube. Diameter of 0.1, length of 3.7. I've got my my calculation here. So 3.142 pi times 0.05, because that is the radius squared, times by the length 3.7 is 0.029 millimeters cubed. I'm also noticing these units here tell you what to do. Millimeters cubed, calculate a volume. Do volume of a cylinder, yeah? And then this minus sign means divide it by the number of hours. So I'm now going to divide it by 3 hours and I get this. I have 2 significant figures. I could have also written 9.7 times 10 to the negative 3. And there we go. So I got all 3 marks. Oh, sorry. Let's go back. 1700 lux, typical on a cloudy day. Calculate the percentage increase in gas production between 1700 and 2000 lux. Okay, 1700, 1.2, 2000, 2.9. So we're going from 1.2 to 2.9. All right. It's more than doubling. Yeah, it's more than doubling. It's over a 100% increase. Yeah. So what we need to work out is what this increase is going from here to here, and then working out what this is of a percentage as a percentage of the original number where we started from, because we're increasing. Yeah. So the difference is 1.7. The original is 1.2. Now this makes sense because we're now going to get a number of over 100%. Yeah, because if it was an 100% increase, it would go from 1.2 to 2.4. Well, it hasn't. It's gone to 2.9. So hopefully that makes sense. And funnily enough, I can cope with this math. So we got those right. Suggest why soda lime was not placed in the respirometer with the seedlings grown in the light. Well, that's because he was measuring the rate of photosynthesis using a respirometer. And if, because, you know, and the soda lime, did they even tell you that the soda lime removes? Yeah, it tells you that the soda lime removes any carbon dioxide. Well, if it removes the carbon dioxide in the air, these poor little blighters aren't going to be able to photosynthesize. So I just put because carbon dioxide is required. And I got it on the notes, all right? This obviously was a good enough wording to get the mark. He made the following claim. The results suggest that in maize seedlings, the rate of photosynthesis only exceeds the rate of respiration when the light intensity is above 1510 lux. Right, so 1510, the bubble doesn't move. And that is telling you that, now this is assuming that the seedlings were exactly the same in these two respirometers. It's absolutely assuming that everything was the same. But that's telling you that carbon dioxide is it's telling you that oxygen isn't being used up and the carbon dioxide isn't being used up. It's telling you that the photosynthesis at 1510, that the rate of photosynthesis is the same as the rate of respiration. So I've written this. Rate of photosynthesis equals the rate of respiration because the distance that the fluid moved equaled naught. The positive figures here, these ones, are saying that it's photosynthesis that is going on. It shows that the rate of photosynthesis is more than the rate of respiration. And these ones are telling you that the rate of photosynthesis is less than the rate of respiration. Now, I went off on one a little bit here. So I got mark 0.2 and mark 0.1 in my first two lines. I didn't get mark 0.3. Yeah, I didn't get mark 0.3. Identify these tissues. Well, this is a liver lobule. This is an islet of Langerhans, these pale cells here of an islet of Langerhans within the acini of a pancreas. This is skeletal muscle and this is cardiac muscle. So hence I got these and I wrote a little bit more than was necessary, but I got all 3 marks. I didn't mark those. Identify and correct any biological errors in the student's summary. Okay, so when the heart rate's too low, the level of carboxylic acid. Carboxylic acid should be carbonic acid in the blood becomes higher than normal, right? The vagus nerve sends action potential. No, the accelerator nerve will send action potentials to the SAN to increase the contraction rate of the heart muscle. The baroreceptors in the walls of the blood vessels then detect the pH of the blood. Now, I think that should be, these baroreceptors should be chemoreceptors so that the heart rate can return to resting. The endocrine system can also change heart rate. Yes, it can. Release of the hormone adrenaline from the adrenal medulla, yeah, that's all okay, causes the smooth muscle of the heart, the cardiac muscle of the heart, to contract more frequently. So I think I got all of those, but obviously I only got the 4 marks. Okay, reflex actions, rapid responses to protect the body from harm. Okay, right. Moro reflex is this reflex of babies that if their head is unsupported, they spread their arms out wide and then bring them together and they cry. Suggest how the Moro reflex helps to prevent harm to a newborn baby. Well, this is the baby trying to grab something to hold on to, okay? And the crying is so that they get attention. Something's going wrong. So, okay, so that is those 2 marks. Describe, and here's the final question, describe the reflex response of a 3-year-old child what the reflex response a 3-year-old child would make to an object moving towards their eyes and explain the advantage of this response. Now, this is what I did. I'd blow it. The cross hasn't come up. Hold on. Don't know what's there. Look, sorry. So this is what I wrote. And I suddenly realized that it was a 3-mark question. I'm thinking, I'm going to be writing forever doing this whole, response, what the reflex action is. I was describing the reflex arc, not the reflex response. And I suddenly realized that the question was actually much simpler. Describe a reflex response and explain why it's to their advantage. So they would close their eyes involuntarily and quickly. Here am I thinking of those words that describe a reflex response, and this would help protect the eyes from damage. Now, do you know, that's funny because I'm surprised I got that mark, because I'm basically saying exactly the same as what's up here, and yet they allowed it here. I'm actually surprised that I got that. I did end up getting all 3 marks. I think it would have been much better to just to have said it prevented damage. Oh, I suppose I did say from damage. Anyway, hey ho, I got the marks. So there is the end of OCRA 2018 paper 1. And I'm really hoping that that has been a useful walkthrough. And I'm hoping that I managed to do that relatively efficiently so that it wasn't too much of a long drawn out process. However, that was clearly a long live stream. Thank you for staying with me, those of you who have. I hope you found that useful, and come back next week and I will be doing the same thing with paper 2. All right, see you next week, guys. Bye-bye.